get started. That way we're not delayed. Good afternoon, everyone. My name is Anil Patri. Hello? Yes, you can. We have this session is on clarifying education and workforce training needs in nanomedicine and targeted drug delivery. As you know, this is a fast emerging field of nanomedicine. There are not a lot of new technologies that come through, a lot of new instrumentation. And the workforce training is very critical because a lot of these new instruments, they are easy to use. You press a green button, you get a number, and you have to actually analyze and you understand the technologies. But that is on the physical characterization end. But then when you go to in vitro, in vivo studies, as you develop the nanomedicines, there are more challenges. And so workforce training is very critical, especially when it is a multidisciplinary training, when training has to be thorough in one area and then expand into other areas. So we have an excellent panel of speakers today. And so the first speaker is Dr. Andrew Owen from University of Liverpool. He's going to talk about nanomedicine, a clinical pharmacologist's view of nanomedicine regulation. Dr. Owen.
Thanks very much, Anil. So this is a huge area to cover in a 15-minute talk. So what I'm actually going to do is I'm going to try and use the next 15 minutes or so to really underscore and highlight one area where I think there's a real training need, and that is in the use of physiologically based pharmacokinetic modeling, really at all aspects of the process through development and through into post-marketing areas. So I'll start by underscoring the importance of pharmacokinetics itself. This is a grossly oversimplified pipeline of development of a medicine, any medicine, including nanomedicines, where it starts with lead selection, moving through preclinical development into phase 1, phase 2, phase 3 clinical development, all the way up until launch, and then the era post-licensing, where clearly there's still often a lot to be learned about a particular medicine and its use. So where is pharmacokinetics important in this? Well, really across the whole range of these activities, pharmacokinetics is considered, and it's considered because it's so important. So it starts very early on with early in vitro understanding of drug disposition, and this can include things like understanding drug transporter interactions and their role in absorption or clearance, for example, of the material or the drug. Equally as important, drug metabolism, defining what the major metabolic enzymes contributing to variability in a drug are, and then moving through that into in vivo preclinical validation, specifically focusing on pharmacokinetics. And this includes getting an early indication of oral bioavailability, for example, for an oral medicine, factors which influence clearance, dose linearity, and all these types of things which are important for the development, and then moving through that into early characterization of the exposure-response relationship. Moving out from that and moving into clinical development, then there's clearly a need to start thinking about what the human doses might be, and at that stage, there's a need to think about human dose prediction. Later on in development, then you move into the human pharmacokinetic studies, again looking at bioavailability in humans, how good your predictions were, looking at clearance, dose linearity often, and then looking at the pharmacodynamics later in clinical development, including the exposure-response relationship, which includes an understanding of the efficacy as well as the safety of the medicine. Pharmacokinetics also plays a hugely important role post-licensing. It's really an ongoing process with a medicine. And it feeds into clinical management decisions as well as optimization of therapy for individuals, particularly in chronic disease. And this can include things like drug-drug interactions, whether there are PK differences in special populations. In some cases, and I'll show you an example of this, in dose optimization where post-licensing there's a need to maybe reduce the dose of a particular drug. And also in understanding pharmacogenetic predictors, for example, in terms of the variability and the response. Now, physiologically based pharmacokinetic modeling can have a hugely important role across these different areas. And indeed, the bold-faced points there on this slide are where physiologically based pharmacokinetic modeling actually specifically features in various guidance from the regulatory authorities. So already an acknowledged role of PBPK modeling in terms of conventional medicines. This is an example of a project we've been involved in recently. It's not a nanomedicine, but it's an example of how when you get PBPK modeling right, it can be very useful in terms of dose optimization. And this is in the post-licensing arena. So back in, I think, around 2012, we actually conducted PBPK modeling for an antiretroviral drug, which is a drug called efavirenz. Essentially, around that time, there was a lot of conversations happening about whether the dose of efavirenz, which was dosed at 600 milligrams daily to patients, was too high and unnecessarily high to fully suppress viral replication. So there's a lot of conversations around whether that dose could be reduced to 400 milligrams daily instead. We did a lot of modeling on efavirenz pharmacokinetics. We did the IVIVE, or the in vitro in vivo extrapolation, which is basically the PBPK modeling, coupled with population PK modeling for the 600 milligram dose, and estimated, using the extrapolation data from the model, what the pharmacokinetics would be like if the dose was lowered to 400 milligrams. After we'd done this, there was a trial initiated called the Encore 1 study, which actually validated in patients at a multi-site center what the pharmacokinetics and response to the drug was at 400 milligrams versus 600 milligrams. Our group did the pharmacokinetics in the Encore 1 trial. And this is a paper we published recently showing how good our predictions from 4 or 5 years ago were to the actual outcome from the Encore 1 trial. And you can see that we got very good agreement across the various pharmacokinetic components between our predictions, our prior predictions using PBPK, and what we actually observed within the trial. So this can work. What is PBPK modeling? Well, I'm going to give it in a snapshot in very simple terms. Basically, PBPK modeling is the use of in vitro data and physiochemical descriptors of the drug or the molecule or the nanomedicine to model the distribution of the drug around the body. So effectively looking at movement of the drug through the gut into the systemic circulation, distribution around all the different tissues, clearance through the liver, through the kidneys, and then as a result of that, you get an estimation of the pharmacokinetics. So you get clearance, you get volume distribution, you get bioavailability, and if you've got robust pharmacodynamic data, so if you've got activity data, or if you've got safety data for a concentration-dependent toxicity, then you can actually incorporate that into the model as well in order to get a feel for what dose you need in order to stay within the therapeutic window. Of course, it's actually much more complicated than that. I'm not going to go through this in detail, but we're doing all the PBPK modeling in Liverpool using SimBiology and MATLAB, and this just gives you an idea of what the fundamental model looks. And essentially, just to cut a very long story short, what you've got is you've got 300 differential equations which describe the passage of the drug and or metabolite in and out of each compartment within the body. And to go back here, what you're using to model these different things are various in vitro data. So for example, the role of metabolism enzymes, drug release rate if it's a nanomaterial, passage across intestinal barrier if you're looking at an oral medicine, and it's not difficult to see that you might want to incorporate things like macrophage uptake when you're talking about a nanomaterial. It is important to state up front, predictions are only as good as the depth of the fundamental knowledge that you have, as well as the appropriateness and the quality of the input data that you have to put into the model. And there's an old adage which states all models are wrong, but some are useful. And we've got to keep that at the front of our minds. So I think it's important to say fundamental knowledge is key to this pharmacokinetic prediction. When you're talking about absorption, distribution, metabolism, and elimination, you need a good understanding of the anatomy, the physiological, and the molecular aspects of what are important. And only when you have that, and bear in mind that this knowledge builds up probably over decades before you start using it in this context. Once you have that, then you've got a rational basis for the development of the in vitro tools which you need to get the output data for a robust prediction of the pharmacokinetics. And then you've got a better PK prediction which comes out through your model. So what are the differences in the absorption, distribution, metabolism, and elimination for small molecules and nanoparticles? And I'm just going to go through, so for example, oral bioavailability, distribution, and clearance of small molecules is given mainly by the physiochemical of that drug, but also the affinity for drug transporters and metabolic enzymes. But obviously, many of the nanocarrier systems are very complex, often multifaceted, and we need to think about information on all components of these nanocarrier systems in order to get a robust prediction of the response, or of the exposure. So for nanomedicines, there's a need to consider these drug-specific factors for the drug which is loaded within the material, but also you need to think about the physical structure in terms of the nanoparticles themselves, so the composition, the size, the zeta potential. There's also a whole set of other molecular, physiological, and anatomical factors that you need to consider. Some of these, for example, are the uptake by the reticuloendothelial system, which clearly impacts the clearance of nanomaterials, may be beneficial in certain diseases for which the target is a macrophage. But we need to collect data on that to robustly model. We need to think about how the macrophages are infiltrating the different tissues so that we can model how the nanomaterial goes with those macrophages. Apparent permeability through advanced intestinal model systems, and I'll talk about that on the next slide. We also need to think about capillary fenestrations, for example, and how they restrict passage in nanomaterials. Think about the degradation and the drug release in biological fluids, as well as the impact of nanoparticle protein corona, which can also influence many of these other factors. So we need to better tools, better knowledge of these different processes when we're considering PBPK modeling for nanomaterials. So this is the example for oral bioavailability. I'm sure many of you have heard of CACO2 cells. They've been around for a very long time. They're an industry-used model system for looking at the oral absorption of small molecules. And PBPK modeling all goes back to this original paper which showed the permeability of CACO2s correlated, across CACO2s correlated with human jejunum permeability. And that's CACO2s, by applying the equation of this line to a CACO2 cell data, you could robustly get a prediction of the oral absorption of a small molecule. But what about nanomaterials? Well, for nanomaterials, there's a number of different models emerging. These are triple culture models. They incorporate many of the features of the intestinal tract which are absent from CACO2s. And it's been shown that the triple culture models are much better at showing similar permeability to ex vivo samples, so typically intestinal biopsies, which are assessed in an assaying chamber. So a much better model for nanomaterials is possibly presented by these triple cultures. And that's because, whereas a CACO2 will only capture this intestinal epithelial cell transport, the triple culture models will capture the effect of goblet cells, which are secreting the mucus onto the cells, which can trap nanoparticles, as well as differentiating some of this epithelial cells into M cells, which can be a route via endocytic processes through the intestinal barrier and into the systemic circulation. So we also need to understand the pharmacokinetic, pharmacodynamic response. We've got here a typical PK profile for a once daily drug. We know that above this concentration, you've got the higher potential for toxicity, and below this concentration, we've got insufficient concentrations for efficacy. So we need to keep plasma concentrations within this therapeutic window. We know that many of the safety considerations which are nano-specific, for example, the immunological safety assessments, are concentration dependent. Most of this work has been done by the NCI-NCL in terms of immunology, but this is one paper that has emerged from our group in Liverpool, showing the effect of gold nanoparticles, for example, and this concentration-dependent effect on T cell proliferation in an ex vivo assay. So we can use these in vitro tools to try and define at an early stage what these high concentrations are in order to marry that with the physiologically based PK modeling. So that in a snapshot is what PBPK is and why we need it in the nanomedicine field. But this is really, the y-axis here shows the number of citations per year, and this is from the 1988 to 2016. In green, we've got the total PBPK papers, and you can see that it's now around 140, 150 papers per year for PBPK modeling. But let's look at how many papers are emerging in the nanomedicine field. So nano-specific PBPK modeling. You can see the first one was in 2008, and although it's starting to increase, we're nowhere near the same level as PBPK modeling is being applied to small molecules. So to put that in context, now we're at the place we were with small molecules in the late 80s, early 1990s. So a real need for more PBPK modeling. I'll skip over my summary and conclusion slide because I've already seen it. We are offering a modeling co-service as part of an antiretroviral long-acting medicine development resource program funded by the NIH. The modeling is offered out of Liverpool. So if there is anybody working on HIV, you can access this free facility for PBPK modeling of your nanomaterials. Shameful advert for our new journal, the Journal of Interdisciplinary Nanomedicine, and I'll just leave you with some acknowledgements. Thank you.
The last slide you showed, and we saw the red nanomedicines. The question is, how would you see the line going on? I mean, this and then that. So the question is, why less this year than last year? Because it's July, it doesn't look like. So
that's a great question, Beard. And so my view on that is we're still at a stage now where people like me are at conferences saying we need to do this, right? But the field really hasn't really hasn't really come to terms with using PBPK for nanomaterials. So that's part of it. The real big piece of it, though, is there just aren't that many modelers. This is a very specialized area. PBPK modeling is quite intensive, it's very mathematical, it's interdisciplinary, and there just aren't that many modelers around. So I think there's, and you know, to get back to the reason for this session, I think there's a real workforce training need there for PBPK modeling.
Not fully answered for me,
Okay.
because if there would be a huge amount of products, it would influence this curve drastically.
True, very true.
So we have been discussing in the last days in small circles and big circles, coming to the truth. And the question in such slides are, how true are they? How can we increase? How can we really move? That would be the truth towards having the European Union and all stakeholders further investing. That would be convinced that the science is going on in this field very prosper and very hopeful.
So yeah, I agree with that completely. What I would say is what I would say is it's a catch-22 situation. I think that PBPK modeling can help us get more products. I think that's the point that I'm trying to make. What I'm saying is there's a lot of preclinical data out there. What PBPK modeling can do is it can take that preclinical data, it can take the in vitro data, and it can turn that data into something which can help drive these materials through those early stages of translation where they tend to fall off. So so what I would say is PBPK modeling is a tool that can help us accelerate more of those nanomaterials through to more common use.
I think you're absolutely right, but in praxis, most people think about modeling at a rather later stage, namely when they prepare their submissions to the agencies. True. When they have to present the data. Before that, they do much more kind of phenomenological observations, depending how good their development team is. And I think that's the opportunity where we need to push into the translational space and perhaps offer some more of the insights in the modeling, but also modeling with different materials, which we know, and those of you who just jump from one or the other session, the materials are quite different, and of course the compartmentalization and the whole metabolism or distribution is so dissimilar in so many ways that it's not just like, you know, doing population analysis for a different antibody, just another antibody and yet another antibody. So there is challenge, but there's also opportunity, and I think it's worth thinking how to influence and push it to move some of the early stages further on, and perhaps use the opportunity of modeling simulation to speed up the development.
If you allow one last question, would you not be the expert? Maybe the workflow, the chain of workflow is not set the right way. I mean, if pharmacokinetics comes at the very end, before you go to the regulator, what you told us is you should do it initially to come to products. So you, with your authority, would be able to show us a different flow of steps, and that would be very, very interesting. I think the community in this situation must learn what would be, by experts like you who do this, published, largely published, how would an ideal chain look to come sooner to a finding, translation to a product?
Yeah. Well, we published a number of book chapters and review papers recently in that area. We've got 2 oral nanomedicines in clinical assessments at the moment. We integrated PBPK right at the start of that program in order to get to lead selection. But for those medicines, it was all about PK anyway. The motivation was a PK adjustment, and the nanomaterial was, from a regulatory perspective, quite simple. But that process that we used in terms of getting PBPK in early is something that we partially, at least, validated in the nanomedicine context.